Today, an FDA approval in epithelioid sarcoma, priority review designations in prostate cancer and multiple myeloma, and encouraging findings in lung cancer and pancreatic neuroendocrine tumors. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has granted an accelerated approval to tazemetostat, known by the trade name Tazverik, for the treatment of adult and pediatric patients aged 16 years old and older with metastatic or locally advanced epithelioid sarcoma that is not eligible for complete resection. The approval is primarily based on findings from a cohort of patients with epithelioid sarcoma in the ongoing single-arm Phase 2 study EZH202 trial, which showed that treatment with tazemetostat elicited an objective response rate of 15%. This included a 1.6% complete response rate and a 13% partial response rate. Among responders in the trial, 67% of patients had a duration of response of at least 6 months. Epizyme, the developer of the EZH2 inhibitor, stated that it will conduct post-marketing activities such as clinical pharmacology evaluations to assess the effect of tazemetostat on liver function, as well as the effect of CYP3A inhibitors and inducers on tazemetostat, which will be used to inform aspects of prescribing information. The company will also expand enrollment of Cohort 6 of study EZH202 to include at least 60 patients with epithelioid sarcoma. The expansion is planned to provide more patient experience for potential future inclusion in the label. The accelerated approval of tazemetostat is contingent on the results of a confirmatory trial. In prostate cancer, the FDA has granted a priority review designation to a supplemental new drug application for olaparib for the treatment of patients with metastatic castration-resistant disease who have deleterious or suspected deleterious or somatic homologous recombination repair gene mutations, and who have also progressed on prior therapy with a new hormonal agent. The designation is based on findings from the Phase 3 PROfound trial, in which olaparib led to a 66% reduction in the risk of disease progression or death compared with either abiraterone acetate or enzalutamide in patients with BRCA1/2 or ATM mutant mCRPC. Overall, the trial showed that olaparib demonstrated a 51% reduction in the risk of disease progression or death versus either of the anti-androgen agents in the entire population of patients with HRR mutant mCRPC who had mutations in genes for BRCA1 or 2, ATM, CDK1/2, or 11 other HRR mutated genes. The agency is scheduled to decide on the sNDA in the second quarter of 2020. The FDA has granted a priority review designation to a biologics license application for belantamab mafodotin as a treatment for patients with relapsed refractory multiple myeloma who received prior therapy with an immunomodulatory drug, a proteasome inhibitor, and an anti-CD38 antibody. The application is based on results from the pivotal DREAM2 trial, which showed that belantamab mafodotin elicited an overall response rate of 31% in patients with relapsed refractory disease who received the treatment at the recommended 2.5 milligram per kilogram dose. This included a very good partial response or better in 18 patients. In patients who received belantamab mafodotin at 3.4 milligrams per kilogram, the ORR was 34%, which included a VGPR or better in 20 patients. There were 3 stringent complete responses or complete responses in each cohort. Overall, the median duration of response was not reached. At the data cutoff date, 18 patients receiving belantamab mafodotin at 2.5 milligrams per kilogram and 25 at the 3.4 milligram per kilogram dose had a DOR of at least 4 months. The median progression-free survival was 2.9 months and 4.9 months in the 2.5 milligram per kilogram and 3.4 milligram per kilogram cohorts, respectively. Overall survival data were immature at the time of the analysis. In pancreatic neuroendocrine tumors, a pre-planned interim analysis of the Phase 3 SANET-P trial showed that surufatinib improved progression-free survival compared with placebo in patients with low or intermediate grade advanced disease for whom there is no effective therapy. The trial was completed by an independent data monitoring committee, which recommended that the study be stopped early, as the predefined primary endpoint of PFS was met. ChiMed, the developer of surufatinib, stated in a press release that it plans to meet with the China National Medical Products Administration to discuss preparing a new drug application for the agent in this indication. Full findings of the trial will be presented at an upcoming medical meeting. Previously, a Phase 1b/2 trial evaluated surufatinib in patients with histologically well-differentiated, low or intermittent grade, inoperable or metastatic neuroendocrine tumors. Patients were stratified by pancreatic or extrapancreatic subtype. Results show that the objective response rates were 19% in the pancreatic NETs cohort and 15% in the extrapancreatic NET cohort. The median PFS was 21.2 months and 13.4 months, respectively. The anti-PD-1 agent tislelizumab in combination with chemotherapy improved progression-free survival compared with chemotherapy alone as a first-line treatment for patients with advanced squamous non-small cell lung cancer, meeting the primary endpoint at a planned interim analysis of the Phase 3 BGB-A317307 trial. The analysis, which was conducted by an independent review committee, showed that tislelizumab in combination with either carboplatin and paclitaxel or with carboplatin and nab-paclitaxel crossed the pre-specified efficacy boundary versus carboplatin and paclitaxel alone. Moreover, the safety profile for tislelizumab with either chemotherapy regimen was found to be consistent with the known risks of each study treatment, and no new safety signals were identified. Full findings will be presented at an upcoming medical meeting. BeiGene, the developer of tislelizumab, stated that it anticipates discussing its plans to file a supplemental new drug application for tislelizumab as a first-line treatment for patients with squamous NSCLC with the Center for Drug Evaluation at the National Medical Products Administration in China. This week, we sat down with Dr. Vincent T. Ho of Dana-Farber Cancer Institute to discuss future directions for the treatment of transplant thrombotic microangiopathy.
The question is in regards to what the next 5 years will bring in terms of the treatment of transplant TMA. I think we're in an exciting time right now. For the last 2 decades, and nobody's paid attention to TMA because there was really no effective therapy. With the advent of the complement blockers, and with a better understanding of the pathophysiology of this disease, there's now renewed interest in this realm. There's still a lot of work that needs to be done in terms of tying down a good diagnostic criteria, as well as finding good biomarkers to help us diagnose this disease. We now know there are certain risk factors for this disease. If, for example, genetic testing of patients to look for predisposing genes might help us target certain populations to screen and to use prophylaxis in. And then once we are more cognizant of looking for this condition during transplant, we're more likely to diagnose it earlier in the course by implementing more stringent screening. For example, screening for hypertension, screening for proteinuria, and then being vigilant to the diagnosis will allow us to diagnose this condition earlier, and having tests to look for complements will help us decide who might be the best candidates for intervention with complement blockers or with a drug like defibrotide. So I think now there's a lot of interest in this disease because there's now potential effective treatments, and this would be a major step forward because I think it will help improve our transplant-related mortality, because much of the mortality after transplant from graft versus host disease are probably intertwined with morbidity from thrombotic microangiopathy, which in my experience tends to go hand in hand with graft versus host disease, and graft versus host disease is one of the most common complications of transplantation.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.